This was the first urban environment and the first urban trading place in central Sweden. It's a fairly small island, but if you go there and excavate, there is Viking stuff everywhere. And among these were this burial, BJ 581, that we now just call the Lady Warrior. She was buried with an extraordinary, amazing, fantastic sword, battle dagger, armor-piercing arrows, shield, 2 horses, everything that you would use as a high-ranked and efficient Viking warrior. She was excavated in 1878. She was published over and over again for 100 years as the type example of a male Viking warrior. And I keep saying she because there was an osteological study done on her in the early 2000s, where it obviously proved that it was a woman, but because it was osteology, it was never trusted, right? People wanted DNA. And we did. That's the easiest, the simplest analysis we do on ancient human DNA. You don't need much. You just need enough to make sure whether there was a Y chromosome there or whether there were no Y chromosome there. That's how we find the biological sex. And everything else we do, we need more DNA for. But this we don't need so much. And that is good because DNA is not well preserved on this island. We could do a few other things as well, but due to the small amount of data, we couldn't be too precise. We could compare the DNA we were having from this woman with other published stuff and see that she most likely originated somewhere in Northern Europe. The thing here was that she was female. That actually made this little publication, I would claim, the most read archaeological publication ever written. It bounced up to ridiculous numbers in the Bibliometrics, read by far more than any cancer research or whatever you want. It was a woman and she was buried with a sword, and that was enough to make everybody emotional one way or the other.
So now we have ancient DNA. So what we wanted to do is to establish the phylogeographic pattern prior to domestication. And then we wanted to observe whether this phylogeographic pattern would change over time. And this would give us a hint of things that are related to domestication, evolution. We have specimens that come from Syria, from the Levant, from Egypt, Northern Africa. DNA is really poorly preserved. On top, we will have small bones, not very thick cortexes, not dense bones, so nothing to do with this mammoth. And I agree with Anders, mammoth DNA is ridiculous. This is just 1 million years old, yeah, but it looks like model. So we devised an assay to get the most information at the least cost, because we didn't have a lot of money, and to get the most information out of these terribly preserved bones. And so we combined a multiplex PCR to target many specific SNPs that are adapted to the characteristics of ancient DNA, and then subsequently sequence these PCR products with NGS. And this worked, so we got a publication out, and I will tell you what we published. The earliest occurrence of mitotype 4A star, which is one of the present-day domestic clades or lineages, outside of Anatolia, was found in the Balkans, and it was found in a Neolithic context. So the first Anatolian cats arrived in Southeast Europe with the Neolithic farmers and spread then around the Mediterranean. This is the only conclusions we can deduce from that. So what we think is that cats in the Fertile Crescent developed a commensal relationship with the early farming communities, accumulating the cereals, the seeds, which will attract rodents.
Our skeleton is a record of our life history. Anything we eat or drink has a chemical signal that passes into a system and is recorded into our tissues. So chemical analysis of body tissues can actually trace it back to the signal of the food and water consumed during our lives. We normally use analysis of carbon and nitrogen isotopes in relation to diet, an analysis of strontium and oxygen isotopes in relation to residential mobility and geographical origins. Because our teeth are formed early on in our lives, and because tooth enamel does not remodel after it has formed, therefore, if you break a tooth, that doesn't build back as it happens with bone. If an individual was born and raised in the area where they were buried, the strontium and oxygen signatures in their teeth should be similar to the local bioavailable strontium and oxygen. If we find these signatures to be different, we may infer that these people moved from the site where they were born. Mobility is also relevant in relation to past people economy and in relation to herding practices these people followed. And in relation to this question, we investigated transhumance in South Turkey in the Bronze and Iron ages. We applied the strontium and oxygen isotopic ratio analysis on archaeological animal teeth from the site of Kilise Tepe here. You probably know better than me that transhumance is practiced nowadays by the Yurok people who relocate their animals during spring from the Mediterranean coast to the Taurus Mountains. So what we did was to analyze archaeological animals from the site of Kilise Tepe. We can see that these 4 individuals here, their patterning of the data differs to the patterning of the data we have seen from the precipitation data here. So we may infer that they moved seasonally between higher and lower pastures during the year.
There was a discussion about where did the domestication of horses took place. So we wanted to test the hypothesis, did it take place in Anatolia, and when did it take place? We used sampling from the Neolithic to the Iron Age with a big focus on this specific area. So you will recognize for some of you that went to the field trip, and some that we're going to see also after Atusha. Basically, we looked at 2 aspects, mitochondria and coat color. And the strategy was the same as for the cat, to target specific SNPs in the mitochondria that allows to reproduce the phylogeny that we know from the full mitogenome. So this is what we have with the full mitogenome, and this is what we have with what we targeted. So the idea is to target by PCR, use the next generation sequencing and do multiplex PCR so that we can have plenty of margin. After 2000 BC, we have a major shift in Anatolia and the Caucasus, much larger mitochondrial diversity and a very large coat color diversity of the horses. So what we believe we have here is that before 2000, wild horses, they have a white coat color, and they have a local diversity because they have not exchanged with the horses from the northern part of the steppe that have a different diversity. And we see an import of domesticated horses from elsewhere. And when we look when it happened, it seems like it goes through the Caucasus. So the idea is that domestication took place a little bit more north in the steppe area and was imported in Anatolia around 2000 BC.